So it's great to be here in Michigan. I'd like to thank YQ for extending the invitation for me to come and talk to you today. I've had great conversations with a lot of people here in the department today, and in fact, I've had so much good conversation today, I've barely had time to get nervous about giving this talk. All right, so my talk today, water ferns, ancient wetlands, and a case of mistaken identity. So the first part of this talk is going to be recounting some research that I began as a postdoc while at Cornell University. And I've continued working with those same collaborators as I've begun my faculty career at Ohio University. The second part of the talk is going to be about North American material, and this is a project I've been pursuing on my own. So, all right. So I guess before I begin the South American part of this talk, I'd just like to recognize and thank my collaborators and funders for supporting my work on this Patagonian material. So on the water ferns of the La Colonia Formation, I've been collaborating with Ruben Cunho, Maria Gandolfo from Cornell, Ruben's from Argentina. He works at the Museo Paleontológico Edigio Truglio in Trelew, which is in Chubut province. And also Maria del Carmen Zamaloa from the Universidad de Buenos Aires. And so my involvement in the project has been supported by 2 NSF grants to help pay for my postdoc and also for my travel down there, and also by some funding I got from Ohio University. So here we have Ruben staring off in the distance, contemplating, I guess, the sediments in the field. Alejandra with the GPS. Here's myself and a student, Julieta Gallego. I went out to La Colonia in 2010. Unfortunately, it wasn't a plant fossil collecting trip. We were there to do some stratigraphic work, but it was still nice to see the formation that these fossils came from. And then over here, there's just a group of us who were on that trip, and I got to wield the Jacob staff to help measure the section. All right, so like I said, the first part of this talk is on Patagonian material. So where are we in the world? Well, unfortunately, I guess this map of South America today is a bit small, but we're down here in Chubut province, which is in the southern part of Patagonia, Argentina. So this is a blow up of the province of Chubut. Trelew, this town here near the coast, is where the museum is located that our expedition was based out of, and it's also where the fossils are permanently housed. And then the study area is over in this region, so I guess best characterized maybe as the middle of nowhere. Down here is a blow up of that. So this just shows some local waterways, a local roadway, and then the 3 localities that have yielded plant fossils. So these are Cerro Bosta over here, Cañadón del Irupe, and Quebrada del Helecho. So I guess sort of like Fern Canyon or something like that, an apt name. Here is South America at about this time. So this is a reconstruction. The La Colonia locality still would have been relatively high latitude down there. All right. So here are a couple of stratigraphic sections. Basically, the plant localities are correlated here. So this is a section summarizing the formation at Cerro Bosta. This is a section summarizing the formation, the La Colonia formation at Cañadón del Irupe and Quebrada del Helecho. The section that yields plants is thought to represent a freshwater lagoon type environment. Now you may be able to see there, or you may not, because this is kind of a detailed illustration. There are a bunch of other levels in these formations that yield different types of fossils. So these little bones, they look like diagonal lines, are vertebrate fossils, and then we also have invertebrate fossils as well. So the La Colonia Formation is really best known for yielding vertebrate fossils. And probably the most famous of these is illustrated up here. This is Carnotaurus, which is an Argentinian carnivorous dinosaur. But also other animals have come from here. So plesiosaurs, turtles, snakes, mammals and birds, and I think lizards as well. So there is quite an interesting vertebrate fauna. But the plants that we're studying haven't really been found in conjunction with vertebrates. You may also notice, if you're able to read it, that there are sort of different phases in the formation. So the plants are in a freshwater phase, but sometimes there's sort of an open lagoon. This is near the coast, so maybe we get saltwater incursions and things like that. And this, again, is us measuring section in this formation. All right, so here's the La Colonia formation today. It's pretty dry. It's a pretty desolate landscape. Here's our reconstruction of the La Colonia formation in the past. So these are the freshwater lagoons. This is Cerro Bosta and Cañadón del Rupe and Quebrada del Lecho. So we have some floating aquatic plants in there, some aquatics that are rooted in the substrate. There's a surrounding vegetation, so this is thought still to be a wet substrate, so maybe a marshland or wetland environment that's surrounding the lagoons and shedding some of the plant organs into the water where they're preserved. And then finally, we have a more upland vegetation that is represented by pollen. All right, so what do we find in the La Colonia formation in terms of plants? Well, I'll break it down into three components. So in the upland component, and this is represented by pollen that's presumably been produced and then brought in by wind from plants growing in the surrounding area. We have conifers, so Chiralopediaceae and Podocarpaceae are two of the conifer groups we see. Nothofagaceae, so Nothofagus, of course, is the famous southern beech. Something expected in the southern hemisphere. We have Proteaceae, which is another angiosperm family, and then other angiosperms. In terms of the wetland and marsh environment, this is represented both by pollen and also by leaf macrofossils. So we've got a selection of dicot type leaves here. We don't know really what angiosperms these go with, or we haven't really identified the angiosperm groups that yielded these fossils yet. Here we have something that looks like it may be a piece of a cattail leaf. Up here we have a little bit of a conifer, not necessarily that well preserved. And then we also have these nice fern leaves. So we think this is from a Dixoniaceous fern, basically a type of tree fern. And so this would be a modern day tree fern that presumably would be somewhat related to this fern we're finding in the La Colonia formation. All right. We also have things in the lagoonal environment. And that's what I'm interested in and what I'm mostly talking about today, the water ferns, but we also have other things. So we have some algae, which are depicted here. We have the lotus. This is Nelumbo puertae. The species name is for one of the staff at the MEF who's been really integral to our field work. And this was described in 2005 by Gandolfo and Cunha. And so this is the leaf, which would be peltate, and then here we have a fruiting structure that presumably went with it. And then we've got this other floating aquatic monocot, Aquaphyllum. And this was described by Gallego et al. Just recently, 2014 or 2015. So presumably it would be maybe a pistia-like plant, so that would be the water lettuce. So we have rhizomatous aquatic macrophytes and floating aquatics and also some algae. All right, but what I'm here today to talk about are water ferns. And we also find water ferns in this formation. So what are the water ferns? Well, the water ferns actually form a clade of two families of ferns, and molecular phylogenetic analyses suggest that these families are sister to one another. So these are the Marciliaceae. The Marciliaceae today include 3 genera of ferns. They are characterized by having rhizomes, so rhizomes are horizontally growing stems. These ferns are rooted to the substrate, and they have simplified leaves. So the leaf structure varies from Pileolaria, which the leaves almost look kind of grass-like. They're basically reduced to a leaf stalk. And the more complicated structure is found in Marcelia. These leaves almost look like floating four-leaf clovers, so that's where the fern gets one of its common names, which is the water clover. Over here we have Selviniaceae. So Selviniaceae includes two genera of floating ferns. So we have Azolla here. This is the mosquito fern. And then we have Selvinia. So in addition to being aquatic, one of the interesting things about these ferns is that they're heterosporous. So Marceliaceae and Selviniaceae form a clade within the larger group of leptosporangiate ferns. Leptosporangiate ferns represent most of the fern diversity we see in the world today. Most of these leptosporangiate ferns are what we call homosporous. So, in case you don't remember your plant life cycles, I just wanted to go through this quickly to let you know what the difference is. All right, so in a homosporous fern life cycle, what happens? Well, we have what we call the sporophyte. This is the diploid generation of the fern. If you think of a fern, this is what you would think of as the plant itself. This sporophyte has specialized structures known as sporangia. And meiosis takes place, or cells in the sporangia undergo meiosis to produce haploid spores. So we all know ferns produce spores. The spores are released into the environment, and they germinate to produce a gametophyte phase. So this is the alternation of generations life cycle in plants. And the gametophyte phase is haploid. In ferns, it's typically potentially bisexual. This goes along with the homosporous life cycle. So potentially the gametophyte could produce both eggs and sperm. So the gametophyte produces gametes. The gametes combine during fertilization to form a zygote. That zygote then grows into a young sporophyte. And finally, we get the fern that we see growing out in the environment today. Heterospory is a little bit different. So in heterosporous ferns, again, the structure or the organism we think of as the plant is the sporophyte. So this is the diploid generation. So this sporophyte also produces sporangia in which meiosis occurs. And I'm sorry I didn't have some fancy spore pictures for you for this one. But basically, there are two different types of sporangia, and they produce two different types of spores. So we have the megaspores, which as the name suggests are the larger spores. The microspores, which as the name suggests are the smaller spores. These spores germinate to yield two different types of gametophytes, and the gametophytes are unisexual. So this is the haploid phase of the plant life cycle. So we have the megagametophyte, which produces the eggs, and the microgametophyte, which is going to produce the sperm. These gametophytes develop within the wall of the spore. So unisexual gametophytes developing in the spore wall goes along with heterospory. So they produce the gametes. Gametes fuse during the process of fertilization to form a zygote. The zygote then becomes the young sporophyte, and finally we get our fern once again. So presumably, heterospory in these plants would be an adaptation to living in the aquatic environment. They release these spore structures, they have the gametophytes within them, fertilization takes place relatively quickly, and the young sporophyte develops, or the young sporophyte begins to grow in the aquatic environment. Heterospory is something that has evolved a number of different times in vascular plants. So we also see it in lycophytes, we see it in some extinct horsetails, and most importantly, we see it in seed plants. All right, so another interesting thing about the water ferns, or somewhat unique characteristic, is that their sporangia, so their spore-producing capsules, are enclosed in structures known as sporocarps. And the things we call sporocarps in these two different groups, by the way, aren't exactly the same thing. But we don't need to worry about it too much for the purposes of the presentation. So up here we have Azolla And we have some sporocarps with microsporangia in them. Down here, we have a sporocarp of Marcelia. In Marceliaceae, the sporocarps are these really thick-walled structures. So this has been cut longitudinally. Unlike with Azolla, you wouldn't really be able to see the sporangia within the sporocarp if it were whole. And then here's some sporocarps of Regnolidium. So these sporocarps open up to release the sori and the sporangia, which release their spores. All right. So within the La Colonia formation, we actually have a number of different water ferns. And we have two of the family, well, we have both families represented. So within Salviniaceae, we appear to have just the Azolla lineage represented. So this is just a little bit of information about modern Azolla. Azolla, sometimes called the Mosquito Fern, includes about 6 species of floating aquatic plants. They occur worldwide, native or introduced. Their leaves, so you can see here, they have these really simple leaves. This plant is actually probably best known because it harbors cyanobacteria in its leaves. These cyanobacteria, I believe, are in the genus Anabina. And these cyanobacteria fix nitrogen, which of course provides an essential element or nutrient to the plant. And so if you look up Azolla, you'll probably see a lot of papers about using Azolla as a biofertilizer, particularly with rice. These have thin-walled sporocarps. So here's an example from a herbarium specimen of some sporocarps with microsporangia in them. And the microspores, so these small spores, occur in clumps, and these are called maculae. So it's just a little bit about the structure of Azolla. In terms of the fossil record, this fern, or things that are thought to be related to this fern, have a fairly extensive fossil record, and it's really bigger than I can get into here today. The record begins in the Cretaceous, so maybe 84 million years ago or older. There are a number of different extinct genera of Azolla-type spores, and there are about 10 macrofossil species worldwide. So it doesn't have an extremely extensive macrofossil record. Most of it's known from North America, so there's been a lot of work done in North America, and we'll see this again. We know a lot about the Northern Hemisphere record. Macrofossil species go from the Maastrichtian to the Eocene. So the Maastrichtian is the very end of the Cretaceous period. And the Eocene is about 35 to 55 million years ago. So these are some examples, not from the La Colonia formation, but you can see there are some really nice Azolla fossils from North America. So what do we find in the La Colonia formation? Well, we find both spores and sporophytes. So these are the fossil spores over here, and for comparison over here, these are some modern spores. So fossil spores fall into 2 genera. This is presumably assignable to Azolla, the modern genus, and this is Paleoazolla. So some things to notice about these are the megaspores, so the large spores. This is a megaspore of modern Azolla here. There's a spore body, a collar, floats, and then this is an indusium attached to the top of the spore. This Azolla spore over here, you can see the body, and these depressions indicate where the floats would have been. So it doesn't actually have floats on it. And then down here, Paleoazolla is a bit different. So you can see the difference in the shape of the spore body, and here are the floats up here. This is a microspore macula of modern Azolla. One interesting thing to note about it is that it has these characteristic structures. Maybe we can think of these as hair-like structures. These are known as Glochidia. And in this case, they have anchor-shaped tips. So you can kind of see that anchor-shaped end on them. That's characteristic. Over here, we have a similar thing from the La Colonia formation. So an Azolla microspore macula. And then here is a close-up of one that goes with Paleoazolla. In this case, there's an anchor-shaped end, but there are also barbs on the side of it. So we have a little bit of diversity of Azolla-like ferns in this formation. Here are some fossils, and I know the fossils maybe don't look like the most impressive things. But if you look closely, here we have a herbarium specimen of modern Azolla. You can see that it's got imbricate, or sort of these overlapping leaves on it. You can see that here if you look closely at the fossil. Here's just a larger fossil, so we can kind of see an axis with branches and leaves. This is a detail of this. And we also have fertile specimens. So we have microsporocarps on this fossil, which is within that circle there. They're a bit hard to see. But down here, we have a herbarium specimen with microsporocarps for comparison. And we also have some megasporocarps. So we have fertile specimens. This is potentially good, because if we can match up the sporophytes, so the fern macrofossils, to the spores, we have a more complete understanding of what the plant was like. Unfortunately, we haven't been able to actually extract any spores directly from the fossils. My hunch would be, just based on the shape of this and also how much this looks like modern Azolla, that it probably goes with this set of spores, but there's no way to say at this point for certain. All right. So the rest of the talk is going to focus on ferns in the family Marciliaceae. So, as I said before, there are 3 genera: Pilularia, Regnolidium, and Marcelia. Our best understanding of the relationships right now is that Pilularia and Regnolidium are sister genera, and Marcelia forms a clade separate to them. Pilularia has about 6 modern species. It's pretty widespread. Regnolidium's got 1 living species found only in South America. And finally, Marcelia forms a clade with about 70 species and is also pretty widespread. You'll probably notice right away that it's pretty easy to tell these genera apart based on the morphology of their leaves. So the leaves of Pilularia look almost superficially, if you look closely structurally, they're nothing like them, but superficially it almost looks like grass. They're just little stalks. Regnolidium has 2 leaflets per leaf. So the leaves are compound and subdivided into 2 leaflets. And then up here with Marcelia, the leaves are subdivided into 4 leaflets. In addition to the vegetative structure, these ferns also have very distinctive megaspores, or these fern genera. So Marcelia is depicted here, Pilularia up here, and Regnolidium down here. One thing to notice about Pilularia and Regnolidium is they both have these extensions coming off the spore. These are lobe structures. One name for them that I'll just use in this talk is acrolamella. These are solid structures, so they can be preserved in the fossil record. But usually they're also associated with a gelatinous structure around the acrolamella too. In Marcelia, we don't have this structure. It's thought to be reduced to just a disc-like form. And you'll also notice in Regnolidium, the lobes of the structure are contorted. In Pilularia, they really aren't. They're more straight. So it's pretty easy to tell these spores apart. So the fossil record of this group is really based a lot on their megaspores. That's where a lot of our understanding of the fossil record comes from. There's a pretty big megaspore record of spores that are thought to be associated with this family. Mostly they're assigned to extinct genera. So we have this extinct genus, Arcelides. It's got this incredible acrolamella on it. And this has never been associated with any type of plant megafossil. So we don't know what kind of plant produced this exactly. But given this structure, we think it goes with Marciliaceae. And then Melospora is this other extinct form. This form actually looks a lot like spores of modern Regnolidium. The structure is a bit more diverse, but you'll see these contorted lobes on the megaspore. There's a microspore record. These are mostly assigned to the genera Cribellasporides and Gabanosporus. And then we have macrofossils. So macrofossils can consist of leaves, sporocarps, or whole plants. The oldest macrofossil, so maybe competing claims for this, Recently, this thing was published, Flabelliopterus mei from China. This is Triassic age. I'm not really convinced by it, and I'm not going to get into it. But prior to the publication of this, the oldest fossil was thought to be near the Jurassic-Cretaceous boundary, and it's assigned to the genus Regnolites. So it looks a bit like Regnolidium. All right. The macrofossil record of Marceliaceae has historically been pretty troubled. So even as early as the early 20th century, A.C. Seward was writing about the fossil record of this group, and he didn't think most of it was even worth mentioning. So that suggests he just dismissed it. In 91, Margaret Collinson wrote that no vegetative fossils can reliably be assigned to this family. Well, since then, a number of fossils have been published, and we found some in the La Colonia Formation that are pretty convincing. So here's an example. Up here are macrofossils of Regnolidium that we found in the La Colonia Formation. You'll notice they consist of leaves with 2 leaflets, like modern Regnolidium. The venation pattern is dichotomizing with a marginal vein, which is characteristic of Regnolidium. You may notice that this looks like it has teeth, so these kind of dentate margin, whereas the Regnolidium on the left doesn't. But that can be a characteristic that's influenced by the environment. So it's not necessarily a big defining characteristic. Down here is a megaspore of modern Regnolidium diphyllum. Here is one of the megaspores we find in the sediments co-occurring with these macrofossils. It looks almost exactly the same. And this is assigned to Melospora lobata. And then this is the microspore, Cribellasporides. This also looks similar to modern Regnolidium microspores. So, the fossil record of Regnolidium itself is very, very sparse. This is it. These are fossils that have been actually assigned to the genus Regnolidium. So we have a Sporocarp here from the southeastern US. We've got these leaves from the La Colonia Formation. There's one other occurrence of leaves way out here in Asia in the Eocene. And then we've got a few spores. And these are Cenozoic as well. There is sort of a question, though, because there is a pretty extensive record of Melospora lobata in the Cretaceous. And the question is, was Regnolidium producing this in the Cretaceous as well? And so far, we don't have a lot of evidence for this outside of the La Colonia Formation fossils. All right. The other member of Marceliaceae we're finding in the La Colonia Formation is this. This is Mirasolita irupensis. And this one has a venation pattern that looks like the venation of modern Marsilia. So the veins dichotomize, they branch, the anastomose, they come together, and they kind of form these spindle-shaped or fusiform openings. So this is a very characteristic venation pattern for Marsilia. The difference is we've only been able to find 2 leaflets per leaf. And also, the leaflets in this case have long stalks and are kind of cordate or heart-shaped at the base. Whereas with modern Marsilia, they're wedge-shaped and there isn't really a long stalk. So we decided to put this in a separate genus. We also have sporocarps associated with these leaves. So here are the sporocarps up here with one of these leaves. Down here, we have a Sporocarp compression. And you can see here, side by side, this is a longitudinal section of a Sporocarp of modern Marsilia. The shape is fairly similar. It looks like in the fossil, these dots are probably the sporangia or spore-producing capsules. We haven't been able to prepare any actual spores out of this, though. And then down here, this shows the thick spore wall, which is probably somewhere in here. Here's the thick spore wall of a modern Sporocarp. And these fossils are also associated with Melospora. So maybe this plant was also producing Melospora-like spores. This suggests that maybe this is a plesiomorphic spore morphology for this group. All right. So working on the South American material, I kind of got a tip off that there might be some more material in North America, and specifically from the Green River Formation. So I started looking more into the fossil record of Marcelia itself. So remember, Marcelia is this form that has 4 leaflets per leaf. It looks like a clover. The megaspores don't have much of an acrolamella coming off them. They're just kind of flat on the end. The sporocarps are somewhat bean-shaped. They can have these kind of points on them that are known as teeth, and they can have a stalk that's fused to the end of the sporocarp that's known as a raphe, but not all of them have these features. So I wanted to look, let's see, yeah. So I wanted to look a little bit more into the fossil record of this group. So what we know about the fossil record of Marcelia itself is currently fairly sparse. We have very few records. There are some megaspore records. These are only Oligocene to Miocene in age. They come from Eurasia. So there aren't a lot of records of this group in the Cenozoic, although the fern is pretty widespread today. What's really kind of difficult is the record of Marcelia-like leaves. This is part of the troubled macrofossil record of Marceliaceae. So when I began looking into this, there were maybe 2 species and 1 report that kind of fit the morphology of Marcelia-like leaves. And these extended from the late Cretaceous up to the Eocene. So we had a couple of Cretaceous reports and 1 Eocene report, and they're known from North America and Europe. So far, there's no confirmed sporocarp record of Marcelia, which is sort of interesting. I mean, in general, the absence of much of a sporocarp record of Marceliaceae is fairly interesting because these plants live in aquatic environments. So they're environments that should be amenable to preserving material, and also the walls of the sporocarps are fairly thick. So it's kind of this interesting phenomenon that we don't have much of a sporocarp record. All right. So it turns out that there are Marcelia from the Green River Formation of the western United States. So the Green River Formation is very well known for its fossils. They're very beautiful. This is a fossil alligator or crocodile. My apologies, any vertebrate paleontologists. From Fossil Butte National Monument, there are all sorts of beautiful gars and especially fish that come out of these formations. But there are also some plants. So the Green River Formation actually preserves 3 ancient lakes, which are shown here in Utah and Colorado and Wyoming. So there's Fossil Lake, Lake Gossiute, and Lake Uinta. So the Marcelia fossils all come from Lake Uinta from a plant fossil bearing member known as the Parachute Creek member of the Green River Formation. This is Middle Eocene. So I've basically found, or in collections, I've found about 7 leaves or leaflets from 5 localities. So these have all come from the Florida Museum of Natural History and also the University of California Museum of Paleontology. And I've looked into other Green River collections and I've asked around and come up with nothing. So they're very rare. All right, and this is what they look like. They couldn't be more perfect. It's just beautiful material. So this is up here, this is a leaf. You can see that it has 4 leaflets. They're somewhat wedge-shaped like modern Marcelia. This would be the petiole, or if you like fern terminology, the stipe, that would have connected that leaf to the stem. Down here we have a leaflet with this beautiful venation. And here we have the veins coming together in a marginal vein, all characteristic of modern Marcelia shown here. It's almost like we took this herbarium specimen and just pressed it into this rock. So, petiolate leaves with 4 leaflets. They have the characteristic dichotomizing and anastomosing venation of modern Marcelia. There's no question that this has the vegetative features, at least, of Marcelia. We still have no spores or reproductive structures to go with it. So while I was in Florida, I also decided to look into another taxon. And this is the taxon Marceliaceae phyllum John Hawley. So Marceliaceae phyllum John Hawley was originally described by Skog and Dilcher in 1992, and it's supposed to be the oldest record of Marcelia-like plant. It was originally described as Marcelia. It didn't have any spores with it or sporocarps, and so it was eventually transferred to this catch-all genus Marceliaceae phyllum, which is supposed to encompass any vegetative structures that look like they come from Marceliaceae. So this is actually the type species of that genus, so it is a very important plant. It includes rhizomes, so you can kind of see a rhizome here, that horizontally growing stem. The rhizomes are rooted at the nodes. This is as described by Skog and Dilcher. The leaves are compound, and the leaflets are basically palmately compound. So again, like modern Marcelia, the leaflets are kind of at the same level. There are teeth on them, venation reticulate with a marginal vein, and maybe Regnolidium-like spores. So here's a leaf of Marceliaceae phyllum John Hollii compared to a leaf of Marcelia over here. And just to compare, these are some spores that come from the Dakota Formation. So the Dakota Formation, this is late Cretaceous in age, is the formation that yields Marceliaceae phyllum. These megaspores also come from the formation. They're assigned to Melospora. So these are megaspores that look similar to the megaspores of modern Regnolidium. So this is a very interesting phenomenon. You have Marcelia-like leaves with Regnolidium-like spores in the Cretaceous, and here's a Marcelia leaf with a modern Marcelia-like spore in the recent. So this has potential implications for us understanding the character evolution of this group. And specifically, was the evolution of the characters of the sporophyte, so the plant, decoupled from the evolution of the spore morphology? So this is one potential way to kind of resolve this. We have these Melospora, which are basically these Regnolidium-type spores appearing way back in the Cretaceous. They appear first in the fossil record. So if we take a literal reading of the fossil record, we can think, well, maybe these evolved first. Maybe this is the plesiomorphic or primitive type in this group. Furthermore, if we're finding them with plants with Marcelia-like leaves, this would also suggest, oh, well, maybe the spore form is the plesiomorphic type and the leaf forms sort of separated or developed separately before the spore forms, I guess, diverged, is what I want to say. So there's some suggestion here from the fossil record that these Melospora Regnolidium-type megaspores are the plesiomorphic spore types for the group, and then that the Marcelia-type megaspores and the Pilularia-type megaspores may have evolved separately from these spore types in the respective genera. But I started to notice something odd about Marceliaceae phyllum when I was looking at it, and that's that I didn't think that it looked that much like Marcelia or necessarily even that much like a fern. So there were some weird features. So here's one of these supposed leaflets. It looks like the ends of the teeth have kind of been nipped, each one very regularly. They've got sort of these divots in them. If we look down here, here's a close-up. Here's one of the divots in the apex of a tooth, and the tooth almost looks glandular, almost like something I'd expect to see in an angiosperm tooth. Here for comparison is a leaf of modern Marcelia. The leaflets on modern Marcelia can also be toothed. The development of leaf teeth is a characteristic that is influenced by the environment. So it's considered to go with the leaves having developed under more terrestrial rather than more aquatic conditions. You'll notice that the teeth in this are very irregular compared to this, where the teeth are very, very regular. And also down here, the venation is a little bit irregular. In Marcelia, those veins form these really nice fusiform or spindle shapes. They aren't in this leaf. And finally, there were a couple of things about these supposed compound leaves I noticed. One was that I didn't see any that had 4 leaflets, which would be characteristic of Marcelia. And if you look closely at this, you see 1, 2, 3 where would the fourth one be? It doesn't look like there's any space for a fourth leaflet. Now, this could be due to the way the leaf was preserved. Maybe it's taphonomic, but maybe it's real. The other thing about this is there's this structure in the middle. There's this weird little bilobe thing. And I thought that's odd, but what is it? So when I got done looking at this, I thought, well, I don't think this is Marcelia, but I have no idea what it is. And I'm not going to say anything about it unless I have some sort of alternative hypothesis. And so this is where sort of the paleontology gods, I guess, smiled upon me, because I was pulling drawers at Florida, which is where I was looking at these specimens. And I came upon things like this. This is Fortuna marsilioides. So these are thought to represent whorls of leaves on a stem rather than a compound leaf. And they're described as having been produced by a dicot-like angiosperm. So not a lot is known about this taxon. It's very, very obscure. It was described by MacGyver and Basinger, I think in 1993, from Canada. I've been looking into it in various collections, so in Alberta and Florida and Denver. So there are more occurrences than are known in the published literature. But despite, I guess, my investigations, I haven't figured out anything much additional about this. I don't know exactly where it goes. It's an aquatic plant. It's got these whorls of leaves. The apical margins have teeth, so you can see them up here. And all of these have a central bilobed structure. So that's another interesting feature they share in common. Now there are some differences. This thing from the Dakota Formation of Kansas appears to have 3 leaves per whorl. The other ones I've seen all appear to have 4 leaves per whorl. So maybe this is a species level difference. Maybe these things look similar, but they're different genera. This particular Marciliaceae phyllum is older than the other Fortuna specimens that I've found in collections. So, and this is just a side-by-side comparison of the leaves. So you can see here, just the shape is similar. The size of these things is actually similar. They're larger than the typical Marcelia type leaves we find in the fossil record, which are maybe in the 1 to 2 centimeter range. The venation pattern is similar. These leaves from the Dakota Formation don't have any little veinlets extending into the areoles, so the openings between veins. And this other Fortuna, this one's from Wyoming, do. But that's a small characteristic, and maybe it's something that just isn't preserved in the fossil specimens. So, I guess my conclusion on this right now is that Marciliaceae phyllum, which was formerly thought to be the oldest record of a Marcelia-like plant, isn't a fern. It's probably an angiosperm, and it's probably something like Fortuna. Should it be moved to Fortuna? I'm not sure, but it can stay in Marciliaceae phyllum for now. So, the record of Marcelia-type leaves then stands at 3. 3 from the Cretaceous to the recent. Even though we have this huge spore record, these are aquatic plants. Presumably they're living in still water, and they're living in an environment where they should be preserved. So we have this leaf type, Marciliaceae phyllum Campanicum. This comes from the late Cretaceous Grunbach flora. This is a flora known from Austria. There's actually quite a bit of material, and Yuri Vacek, and I didn't put the picture up here, was kind enough to send me a couple of pictures of it, and it does have characteristic Marcelia-like venation. Down here, we have another Marcelia-like leaf type. This comes from the Eocene Wasatch Formation from the western United States. Again, it's pretty convincing. And finally, we have this Green River type from Utah and Colorado. So if Marciliaceae phyllum isn't a fern, we can't leave these leaves in that genus because they don't fit. And I would recommend that they all be put in modern Marcelia. Yes, we don't have the reproductive structures, and that would be ideal, but at least for the time being, why erect a new taxon for leaves that match exactly something we can find in the recent. So I thought rather than putting conclusions on a slide, I'd just show some pretty pictures while I talked. These are just various images from our field work in Patagonia. So some nice fossil leaves, I guess sort of a road sign in Patagonia, and some rocks. Down here is from our base camp. So the first night we were there, we had some lambs for dinner. We brought out with us and prepared. Up here is this nice volcanic formation, Piedra Parada. And then here's myself and my postdoc advisor from Cornell, Alejandra Gandolfo, and we're there wrapping fossil leaves with a paleobotanist friend toilet paper. So I guess to conclude, Marciliaceae and water ferns in general have an interesting fossil record, but it's fairly difficult to interpret. We have a lot of spores that are telling us when these ferns arose, where they were occurring, but they're not telling us a lot of information about the morphology of the plants that bore them. And unfortunately, especially with Marciliaceae, for some reason, and I really don't know what it is, we do not have a good fossil record of these leaves. The leaves are difficult to work with. It's probably because the form of the leaves is so simplified that it's easy to mistake or think wishfully that various structures actually represent Marciliaceae. So these are the acknowledgments for the second part of the talk. I've gone to a bunch of collections, taken a bunch of loans, gone to a bunch of herbaria, and I'd like to thank you all for coming to see my presentation and inviting me to speak to you.
Thank you, Liz, for the interesting detective story. So take some questions?
Yes? Oh, wow.
Ooh. Have you been able to take advantage of like micro CT scans to look into the Sporocarp and get more information from the fossils?
We haven't done that with these particular fossils. We did recently, and this isn't published yet because I need to revise it and send it back, but we did recently do this from a fossil from another formation we've worked at down here, Laguna del Unco. And it gave us a little bit of information about the three-dimensionality of it, but actually not a lot of extra information. I suspect with the sporocarps we wouldn't find anything, but it might be worth it to do it on one just to see.
I have a couple of questions.
Sure.
First, do we know the pollen, or are pollen finding the same depth as the tree?
So there is a pollen record known from the Dakota Formation. I'm not as certain about the other localities where it's occurred. I would assume probably some pollen work's been done, but I haven't looked into it that much. I just sort of kind of got off on this Fortuna tangent after I looked at the stuff in Florida.
Because my question is, what's the evidence to draw the conclusion saying that Fortuna is angiosperm rather than Marcelia?
I just think that it doesn't have the characteristics of Marcelia. So Fortuna has been considered an angiosperm. I would presume this is based on the veination type. So you get several orders of veins, and just the way the veins are organized and the way the teeth are organized, that seems like the most likely position for it would be in the dicot angiosperms. So I would say that the case that it definitely doesn't match Marcelia is there, but exactly what it is a little bit more open to interpretation.
Because I think right now in the fossil record, so for plants like water, Marcelia is a fern, right? So you can go from Triassic to Jurassic, Cretaceous, Tertiary. You have no problem. But for angiosperm, and right now the psychological barrier, or the psychological threshold in the botanical community is early Cretaceous. If you find anything that looks like angiosperm older than Cretaceous, it takes a tremendous challenge for people to get it published. Usually those two things will be called substance. And so one of the story about Marcelia is that when the people were publishing in the 80s, somebody did a survey of Coronado, Cretaceous formation, northeastern Australia. So they had a Marcelia type fossil. few years later, two other authors look at those and publish a paper in Science and argue that it is early angiosperm. But now with more sort of phylogenetic revision and angiosperm identity is confirmed for that plant. So there is a link between a Marcelia and early angiosperm. Any other questions?
Yes?
Do you have any guesses as to what fortune that might be? I mean, given that it's a good time for early angiosperms, kind of looks vaguely like some inoculation thing that I've seen.
Yeah,
Grow in the water, but
yeah. Well, some ranunculation things do grow in the water, and there are other aquatics known from Dakota, so it's definitely a possibility. But this is something where I think it'd just be time to go to the herbarium and start kind of looking through potential groups and seeing if anything matches. But so far, I don't know of anything. Oh, that structure in the center? So I don't know, but a lot of the stuff has been collected by Gar Rothwell and Ruth Stockey. And so I've been talking to Gar about it as well, because he kind of told me we had this huge collection in Alberta, so I had to go look at it. And he thinks that maybe it represents an apex.
That's what they look like.
Yeah.
Yeah, like off the top of my head, you would think it's a branch snapped it through it from mud like that. Sometimes it's super difficult there.
Yeah, because I was talking to him about, well, why do you think this is a whorl and not, a leaf whorl and not a compound leaf, like it would be in Marcelia, and that was his response. And it looked like in that one specimen, maybe it's got some underwater leaf form too, which is kind of interesting. Yes?
Hello. Oh, I was wondering if it's possible to look at these plant fossils in conjunction with animal fossils, and if that would be able to help you identify the plants, and also provide information about how wetland communities have changed over time.
I don't think it would help us with the plant identification, but I do think that it would help us get a better understanding of what these communities and ancient environments were like. So there are definitely animal fossils from La Colonia. There are definitely animal fossils from Green River that could be, I guess, harnessed to help understand these environments. But so far, I don't know how much integration has actually happened. I know with La Colonia, not a lot, but it's still kind of in the early phases of working on it. So probably that'll come eventually. We did kind of have a little section in one of our papers about potential herbivores. So.
the Green River Formation that we and its sisters have. Oh, sorry. The Green River Formation is known for its sort of large-scale wetting and drying cycles as a lake that ends up in evaporite deposits. And is that also true of La Colonia? And also, I was wondering, do the modern water ferns live in similarly similar habitats that have extremes between wetting and drying?
All right. So La Colonia, I know sometimes it gets saltwater incursions, and I do think sometimes it's exposed in a drier environment. I don't know how much that relates to the lagoon we're actually finding there with the freshwater plants, but there are different cycles of what kind of, I guess, habitat you're getting there. Modern Marcelia, I think, yes, it is, I guess, subject to varying water levels and maybe drying out as well in more ephemeral wet places or wetlands.